Hello, my name is Pierzchajlo, and this is my presentation for the UGA CAES Undergraduate Research Symposium for Spring 2020. My project is titled Impact of Delayed Feeding on Gene Expression Associated with Nutrient Transport Absorption in Newly Hatched Broiler Chickens. I've been conducting the study in a lab in poultry science for the past couple semesters, and I'm eager to present it today. In early development, chick embryos draw nutrients from the lipid-rich yolk present within the egg. This initial food source sustains the chick for approximately 21 days until they hatch. After hatch, chicks change their food source from this yolk to a carbohydrate-rich feed on farms. Often, this transition is delayed up to 72 hours in commercial broiler chickens, due to different hatch times and logistic impedance. In the hatchery picture on the screen, you can see that some chicks have hatched, while others are still in their shell. It could take anywhere from 0 to 60 hours for all the chicks to hatch. After hatch, the chicks are loaded up onto a transport truck and driven to a farm anywhere from 1 to 12 hours away, sometimes states away. Once they are processed at the farm, they are finally allowed access to feed. This is where we get the 72 hours from. This delay in feeding after hatch could negatively impact the development of many vital systems, including the gastrointestinal tract, although the issue has not been fully researched. Therefore, the objective of the study was to explore how delayed access to feed impacts developmental expression of intestinal amino acid transporters. This was investigated in the jejunum. It's the section of the gastrointestinal tract where most nutrient absorption occurs. You can see it in the image on the left. By measuring gene expression of certain nutrient transport proteins, we can infer the amount of nutrient transport proteins that are produced, and thus the amount of nutrient absorption that occurs. Chicks were fed within 3 hours after hatching, or delayed feed access for 48 hours after hatching. Jejunum samples were collected from 6 birds per treatment group on embryonic day 19, hour 4, or day 1, 2, 3, 4, 6, 8, 10, 12, and 14 after hatch. RNA was isolated from each sample. If you look below, you can see a timeline of the sample days. Here you can see the feed groups and delayed groups and when each group was given access to feed. The integrity of the RNA was verified running ethidium bromide stained RNA on an agarose gel. An example of this verification process can be seen in the image on the left. The RNA was then reverse transcribed into complementary DNA using the superscript enzyme. Finally, the real-time quantitative polymerase chain reaction method was used to amplify and analyze genes of interest. Here you can see an example of the PCR data that we acquired. The data was analyzed using JUMP statistical software version 15. Then we performed a two-way analysis of variance, followed by a post hoc means comparison and a least significant difference test. Differences were considered significant at a p-factor of less than 0.05 and substantial at a p-factor of less than 0.10. The statistical model included the main effects of age and treatment and the age by treatment interaction. Target gene expression was normalized to 18S RNA expression that keeps the results consistent between plates. Data at each age are expressed relative to hatch, which means hatch expression levels are expressed at 1 for easy comparison between genes. If you look at the example below, you can see that the y-axis measures messenger ribonucleic acid levels, while the x-axis measures time. Feed groups are represented in gray, while delayed groups are represented in burgundy. The hatch level is normalized to 1, as explained earlier. You can see there are letters above each bar. Groups without a common letter are significantly different. For example, E19 and hour 4 do not share a common letter, and therefore we would consider them significantly different. For genes that express a treatment by age effect, 2 asterisks represent significant differences and 1 asterisk represents substantial differences. We've also added trend lines to some graphs to help visualize the delayed treatment effect. Body weights were recorded for each sample bird on all dates. Early after hatch, a trend can be seen where the delayed group weighed less than the feed group. From day 8 onward, the difference is significant, and the delayed fed birds were lighter through the end of the study. Getting into the genes, we looked at 6 genes, and 4 showed the main effects of age, while 2 had an age by treatment interaction. For those genes with an age effect, on the next 2 slides, differences are noted with letters. The cationic amino acid transporter 2 functions as a permease that is involved in the transport of cationic amino acids such as arginine and lysine. An age effect is present, but there are no differences between treatments. There is a significant decrease in the total gene expression from E19 to day 14. Where CAT2 shuttles cationic amino acids, the large neutral amino acid transporter 1 functions as a sodium-independent exchanger of neutral amino acids, thyroid hormones, and prescription drugs. It also decreases with age, but the reduction is less extreme. Sodium-coupled neutral amino acid transporter 2 functions as a sodium-dependent amino acid transporter in the small intestines. Levels appeared to increase immediately after hatch, but then return to pre-hatch levels after day 1. No treatment by age interactions were observed, and there was a significant decrease after hour 4. Peptide transporter 1 functions as a solute carrier for dipeptides. Without access to feed, there appears to be a 3 to 4-fold increase from hatch versus only a 2-fold increase when feed is provided. These final 2 genes exhibited an age-by-treatment interaction, meaning expression differed between treatments. Excitatory amino acid transporter 1 is a sodium-dependent amino acid transporter that mediates the uptake of amino acids like L-glutamate and L-aspartate. We first note an increase in expression at day 1 in the delayed group, then decrease expression at day 4 and 8. Finally, increase expression at day 14. The differences at the older ages are reflective of a delayed developmental pattern. The last gene is BO neutral amino acid transporter 1. It is a transporter that mediates reabsorption of neutral amino acids across the apical membrane of intestinal epithelial cells. Its expression does not increase transiently until feed is available, leading to higher levels in feed birds around hour 4 and in delayed birds at day 2 and day 3. A delay in the later increase leads to differences at day 8. From the trend line, we can see that both groups follow the same trend, but it is delayed in the delayed groups and comes after in the chart. So, in conclusion, we propose that 48-hour delayed feeding does affect developmental gene expression of at least 2 amino acid transporters. Gene expression patterns in birds' delayed feed did become similar to those in the feed group with time, though. A long-term reduction in body weight suggests that delayed feeding at hatch could have prolonged effects on growth and metabolism. Further study with a larger sample size and longer runtime would be beneficial. We also plan to run more genes, but we unfortunately had to end the study early due to COVID-19. I'd like to thank my colleagues in the Department of Poultry Science, Dr. Laura Ellistad, the principal investigator for this study, Dr. Jason Payne and Brett Marshall, our collaborators at the USDA ARS and CAES funding. Thank you.